Welcome students, in this video we will discuss about the development of endosperm. You know the total plant kingdom can be divisible into the two groups, the cryptogamia and the phanerogamia, and the phanerogamia again divided into two broad groups, the gymnosperm and angiosperm. Actually, in case of gymnosperm, the endosperm is developed before fertilization, but in case of angiosperm, endosperm is developed after fertilization, and for this reason the gymnosperm endosperm is haploid but angiosperm endosperm is triploid. Okay, now let's see about the double fertilization. And by the process of double fertilization, which is a very important characteristic features of angiosperm, the two important structures are formed. One is the zygote and another is the endosperm. Now, this is the structure of a female gametophyte, and the gametophyte means any haploid multicellular structure is known as gametophyte. So in this female gametophyte, so this cell is called as the egg. This is the largest cell, and there are adjacent another two cells, and these are known as the synergids. Okay, and the synergids and the eggs, these three haploid cells are present towards the micropylar end, right? And in the central portion there are present central cell, and this central cell contains the two nuclei, and these two nuclei are known as the polar nuclei. Just opposite side of the micropylar end there are present another three types of cell, and these are known as the antipodal cell. Now when the pollen tube enters through this micropylar end, the small male gamete united with the egg, and this will form the diploid zygote. Okay, so this will form the diploid zygote, and another, the large male gamete fused with this central cell. There are present two polar nuclei in the central cell, and thus they will form the triploid endosperm. Okay, so the triploid central cell will be formed, and further modification of this triploid central cell will causes the development of triploid endosperm. So this concept is very essential to understand about the development of endosperm. The endosperm in case of angiosperm is formed after fertilization. So endosperm never formed before fertilization as in case of gymnosperm. So the gymnosperm endosperm is always haploid, but in case of angiosperm, since the endosperm are developed after fertilization, so the endosperm is always triploid in case of angiosperm. Okay, and endosperm development occurs prior to the zygotic division. That means when the egg united with the large male gamete, this will form the zygote, and this zygote undergoes the mitosis division. This will causes the further development of the zygote and causes the embryo, the formation of the embryo. But endosperm is formed before the division of zygote. Okay, now this triploid endosperm is a triploid mass of cells. So endosperm is nothing. Endosperm contains many cells, and all of these cells are triploid. Okay, so endosperm is a multicellular structure, and here you can see a seed. Okay. During time of development, you can divide the developmental period into two phases, the early developmental period and the late developmental period. In early developmental period, the embryo actually present inside the seed. Okay, and this seed contains three most important structure. One is the seed coat, another is the embryo, and another is the endosperm. Okay, so these are the position of this three structure. This covering is actually the seed coat, and the red color you can see this is the embryo, and this embryo is covered by endosperm. Actually this endosperm provide nourishment to this embryo, right? So in early development the endosperm is stored in seed. So this is the seed, and in early development the endosperm is stored in seed, but later when development further proceed, and this endosperm stored in the cotyledons. So cotyledons is the first leaf of an embryo, and during the development this cotyledons is formed. And you know, in case of angiosperm there are present two broad group. One is the monocotyledons and another is dicotyledons. Monocotyledons contains only one seed leaf or the first leaf, and the dicotyledons contains the two leaf. So whatever the number is, actually this endosperm is stored in this first leaf of the plant. Okay, so this is known as the cotyledons, and the endosperm is stored inside this cotyledon. Since the endosperm provide the nourishment to this embryo, then what type of nutrient materials are present in endosperm? Actually, there are present carbohydrate, that means the starch is stored in the endosperm, and oil, that means the lipid are also present in endosperm, and the protein. Okay, so all of these three important food materials are stored in the endosperm and which provide the nourishment to the developing embryo. Now we will see the development, the process of development of endosperm, how endosperm is developed in case of angiosperm. Now see, so this is the central cell, and in this central cell the triploid nucleus are present because this is formed due to the double fertilization, okay, and this nuclear undergoes the free nuclear division and this will form the multinucleated structure. So this is the multinucleated structure. This is one nucleus, another nucleus, another nucleus. So there are developed many nucleus because the nuclear division or the free nuclear division is occurred in the central cell, and this will form the multinucleated supercell structure, but this structure is milky or this is actually present in liquid condition. And after the development of the multinucleated supercell structure, the membrane is developed just outside of this nucleus, and this will form a multicellular structure, okay. So these are the multicellular structure. So before the development of this multicellular structure, the multinucleated supercell is present, okay, and after that the cytokinesis is occurred and this will causes the development of membrane and this will form the multicellular structure. And later the cell wall is developed, the deposition of the cell wall around this membrane is developed, and this will, and you know, due to the development of cell wall, the cell become hardens, okay, because the cell wall causes the hardening of any plant structure, and thus by this way, due to the development of the cell wall, the endosperm becomes solid. So endosperm can be present in two form. It may be liquid form or it may be solid form. What will be the example of this endosperm? If we see the coconut, okay, the coconut milk is the liquid endosperm. That is, the coconut milk is multinucleate supercell structure. In the coconut milk, many cells are present, but each cell contains many nuclei, okay. So coconut milk is the liquid endosperm, right? The coconut meat, which is white in color, this is the solid endosperm, okay. So where the cell wall is formed, so this will form the coconut meat, okay. So these are the examples of endosperm. Now the triploid nucleus of the central cell, see, the triploid nucleus of the central cell divide and this will form the multinucleate supercell. So this is the multinucleate supercell, many nucleus is formed here, okay, and so this is the milky consistency, and later the membranes developed around this nucleus. So membrane developed around this nucleus, this will form the multicellular structure, and then the cell wall is developed. So this multicellular structure becomes solidified, the cell wall formation is occurred, so this becomes solid, and this solid endosperm is stored in the seed and later it is stored in the cotyledons, and this is the process by which endosperm is developed in angiosperm. And hope you can understand about the development of endosperm is angiosperm, and in the next classes we will discuss about different type of endosperm. So thank you, and that's it.